Hi, everyone online. Welcome to the AnVIL Community Conference. We are just about to get started. I'm really glad that all of you are here. Let's go ahead. Cool. So my name is Robert Carroll. I'm a faculty member here at Vanderbilt University Medical Center, and I'm excited to welcome you all to the AnVIL Community Conference 2025. It's great to see so many faces here in person. Great to see, I think we had maybe 30 people online right now, and I hope that only grows through the day. So as we're getting warmed up, there's a lot of energy in the room, which I really appreciate. But I want to check in a little bit to see who's here. And so this will be a participatory activity. I know we have a number of students. If anybody's a student, can you please raise your hand? And I'm counting postdocs for this too. There we go. Yeah, there we go. Great. Welcome. Glad to have you. Who here is affiliated with Vanderbilt in some way? Great. Thank you all for coming. How many of you had to travel to get here today? Oh, yeah. Look at that. I mean, that's awesome. I really appreciate it. I know it takes a lot of work to get out there. I've already heard multiple horror stories of delays and issues with the weather yesterday and all that. So I'm so glad you're here. Who in the room is an AnVIL team member? Great. So if you guys look around and you're not an AnVIL team member, these are the people you can ask any questions you have, and they'll know the answers to all of them right off the top of their head. So great. Thank you. Yeah, so we've got a good mix. So, but really, we're all here. All the AnVIL team members are here because we're excited to share what the AnVIL is with all of you. We're excited for the opportunities that it could present for you to learn from what you're doing and how we can facilitate that. And so we're really excited. Again, I know I'm saying it a lot, but we really are. We've got a full agenda today. We're getting kicked off here at 9:30 with the welcome. We'll have a few of my co-leads joining in just a minute, and I'll introduce them briefly. We have a number of invited speakers. It's just a great agenda. So if you have any questions, please feel free to reach out to me. I think you met some of our friendly folks at the welcome desk if you have any questions as well. I know Natalie here is one of the main coordinators. So again, ask us anything. We're happy to help facilitate. I don't unfortunately work in this building, so I can't answer any great hard questions, but we'll get you the answers you need. So across today and tomorrow, we've got 4 invited speakers, all of whom are excellent. Melissa Wilson today from the NHGRI, Paul Harris tomorrow, one of my fellow faculty here at Vanderbilt, of Elisa Manning from the Broad, and then Alex Bick on Friday, who's also from Vanderbilt. So we're great. Thank you all for making the time to come and present. Here's where we are. You all made it here already, so hopefully you know that. This side of campus is the medical center here. We're in across the way is the real hospital space. There's a lot of research buildings and education buildings like this one here to get you acclimated. So what we wanted to start off with a little bit, I know many of you are Vanderbilt affiliated, although I know some of you haven't perhaps even been at Vanderbilt before. I know, Sophie, this is her first time here. It's a crazy world we live in remotely. And there's a university and a medical center. They're now distinct, but the School of Medicine, where we are right now, is there. And we see the storied history here. You can look on the walls. I don't personally know who any of these people are, but there's been a lot of really critical people. Some of them I'll highlight in a minute, but we're glad to have you here. So this building, Light Hall, was named after Rudolph A. Light, who's here pictured, an MD, and he was the heir to a pharmaceutical company, and this was opened in 1977, this building. So a lot of big contributors to making this work. During the Great Depression, Light kept a tab open at a local restaurant for any medical students to eat for free. And this building originally contained School of Medicine classrooms and laboratories, and a lot of that's still true. And they've built up on top of it, so it actually transitions in the higher floors to a different building. But that's fun. And we'll be up on the 4th floor a little bit later. Construction here in the bottom left is the groundbreaking you can see. In the bottom center shows the completed building, the new adult hospital under construction. And the hospital construction is that parking lot in the bottom left figure. So you can see, it's a lot of development that's happened over the years as the city's continued to expand from the downtown. So we're excited that you all are here in specific. A lot of these classrooms are formulated very medical training style, if you're familiar. And that's, with the huge sort of stadium seating here, so everybody can see what's going on. There's a prep area back here. Anyhow, Langford Auditorium, where we'll be, we were in the lobby last night, and we'll be back over there, was also constructed in 1977. Elizabeth Machad Langford is a widow of a mid-Tennessee businessman who had donated the funds towards that construction, and it was named, of course, in honor of her late husband. It's played host to a lot of presidents, vice presidents, CEOs, Billy Joel, and innumerable conferences like this one. So it's a lot of fun. Yeah, again, a lot of history here on Vanderbilt campus. Thinking specifically about some of the Vanderbilt history in the biomedical tradition, on the left we have the Commodore Cornelius Vanderbilt, who in 1873 put in $1 million to fund the university, hoping it would contribute to strengthening the ties that it should exist between all sections of our common country after the American Civil War. Sutherland there next is a Nobel laureate whose work provided the foundation for decades of work in the areas of hormonal regulation, specifically with cyclic AMP and those cycles. Dr. Stanley Cohen, another Nobel laureate, did a lot of work with the discovery and characterization of the epidermal growth factor here at Vanderbilt, with a lot of treatments of different cancer types, lung, breast, head, neck, and GI malignancies. Of course, we can't help but highlight Paul Harris and hopefully terribly embarrass him. But he's also been really instrumental in bringing together the realms of big data and biomedical research for the 21st century, right, with things like REDCap, All of Us Research Program, Research Match, some of the internal things we have with the BioView and the synthetic derivative and the research derivative. Just, we continue to innovate in, I think, really powerful ways. And my department, the Department of Biomedical Informatics, is one of the biggest in the country. It's home to a National Library of Medicine sponsored training program, as well as some other training programs as well. I came up through this training program here actually and decided to stick around because we're doing a lot of cool things. And so we've done things like having a homegrown EHR very early on, which is now retired, but Star Panel was quite a feat for the time. We have an EHR-linked biobank called BioView with a synthetic derivative, and that's a version of the EHR. And now we have cloud research programs that are home to like the All of Us Data and Research Center and our participation in programs like the AnVIL. And so again, we find data really important. We've been doing a lot to build it. We were pioneering things like that EHR-linked biobank. And so it's really exciting to see the opportunities now that that data is more available and to help lose lessons learned from the clinical and translational side. And of course, many of these programs have also been collaborations internally with the Vanderbilt Institute for Clinical and Translational Research, or Victor. And so, of course, really grateful for our team members like Sophie and Queenie from Victor who have been supporting here. And I think we have some alumni in the room too. So just excited for that. And it's really critical to this meeting and AnVIL generally. So I think I want to hand it over. So the other thing that makes this happen, of course, is NHGRI. So we're really grateful for them helping sponsor this meeting and make it happen. And I've got Chris Wellington from NHGRI.
Thank you, Robert. Yes, so from NHGRI, National Human Genome Research Institute, on the funding side of things, one thing we are contractually obligated to make sure is displayed is this graph. So I went ahead and finished that and put it up now. But you can see here, as the cost of sequencing decreased in the roughly 20 years following the completion of the first draft human genome reference, there was a lot of excitement about the new science that was being enabled, what could be done. But there were also those at NHGRI who saw this and realized that we could be working our way toward a future where very few institutions, very few researchers would have the power to analyze the volumes of data being generated. And that was a concern. And that was one of the reasons that AnVIL was launched, was to democratize that, to ensure that we didn't find ourselves in that position. So given that, really excited to be here at the second AnVIL Community Conference, excited to see the people who are here to learn how you're using the AnVIL, what features, what sort of things you're looking forward to doing. I'll just briefly highlight some of the NHGRI extramural staff who are here in case you're looking to find us to ask any questions. Myself, Chris Wellington, the overall lead on the program. People can raise their hands if they want to. Alana, who's the lead on the AnVIL Clinical Resource. Sergio Sen, who's the lead on the outreach side of things. We have Elena, who's on the policy and data release side. We have Mike Lopez, program analyst supporting the project. Believe that we have Arthur Coe. Yes, you made it. Excellent. Good to see. Arthur, who's been working on the imputation panel. Renee Ryder, who's part of the eMERGE program. And Valentina DiFrancesco, the NHGRI Chief Data Science Strategist and the head of the office out of which AnVIL is run. So with that, thank you.
Yeah, we get a lot of great support from our NHGRI colleagues. We're really glad you all are here. So again, I also asked them questions, like Chris said. He said you could, so please do. So I think we've talked a little bit about the AnVIL. Hopefully many of you at least have some idea, but I want to do somewhat more of a deep dive here about what actually is the AnVIL and why we're so excited about it. So the AnVIL stands for the NHGRI's Analysis, Visualization, and Informatics Lab Space. And the whole goal originally when we started about 7 years ago, I think, was to invert the model of traditional model of data sharing with biomedical research, right? And that was a setting where if you went to dbGaP, you would request data, and every person who wanted to analyze it would download their own copy of the data locally to do analysis, right? And that's not something that really scales. As we just saw, more and more genomes, it became harder and harder to actually get that data, to actually have the infrastructure locally for every person to be able to analyze those genomes. And so by leveraging some of the opportunities of the cloud, the idea was that we could bring people to the data. We could bring that analysis to the data, which does great things about supporting sharing, improving security, improving access to the data, making sure that those analyses are able to be run more consistently. I don't know how many of you bang their heads against command lines on servers managed by somebody in your institution hoping things will work. It's a lot nicer in the world of Docker, where those tools can be deployed a lot more easily. So all of those aspects are things that we were planning to do with the AnVIL, and I think we've really been able to do a lot there. And I have to highlight that AnVIL is a secure and federated data and compute ecosystem. You can bring your own data in, you can use data that we provide, and it's an excellent opportunity. Like the NIST 800-171 for analysis of data is something that we're compliant with, right? And I think that's really important. I know this is in the weeds, I think, for a lot of people. I don't know how many of you have filled out dbGaP applications where it's like, hey, yeah, name some IT person who's going to make sure you take care of the data. What does that mean? What's secure, what's not? There are now binding regulations on this. And the easiest way to be in compliance is to go to a platform like the AnVIL. And so we have a number of articles here that you can see if you want to look up more information about that I'll go on right now about being compliant with the updated genomic data sharing policies. Tara and the AnVIL can help with that. There's a lot of ways to do it. Again, it's just the easiest way to have someone else manage the infrastructure to do that attestation. It takes third-party audits. It takes a lot of work. So I encourage you to, if you're interested in pulling these, to look at the AnVIL as a way to do that. Again, it can make things much easier in this space. And so this notice from 2024 is this is the, is an NIH designated data repository. That's the other thing I think about the AnVIL. So if you have an NHGRI award, it's the preferred place, I think, you're right, to submit the data. I think often it's written into RFA. It's been written into RFAs for a number of years now to say, hey, if you're funded, you need to deposit your data in the AnVIL. And so I think it's a great thing, and we're excited for that. It may feel like a burden sometimes, but we're here to help, and we really want to engage. We want this to be a good experience, because that's the other side of data sharing. It's not just getting to use other people's data. It's how do I share that data and making it useful. So again, we are that designated data repository, and we are, I think, the best home for this NHGRI data. And there was a new award, our award, the AnVIL Clinical Resource. Part of that is to help make the AnVIL more friendly to the clinical side of the data. So it's not just a genome-focused platform, but there are more tools. And we'll talk some about that later. AnVIL itself, though, gives you access to 600,000 genomes, nearly 7 petabytes of data across some of these key consortia and data resources, right? It's the place to get GTEx data now, right? It's a place for the CCDG, the Centers for Common Disease Genomics, for eMERGE, for HPRC, for the Centers for Mendelian Disease Genomics, as well as things like Gregor. So new consortia as well are bringing their data in. And so there's so much data here. It's so powerful, such an opportunity. And you can go to anvilproject.org to find more of this information. This is specifically on the overview page there. It's just exciting. Like it's a ton of data. I know here at Vanderbilt, we have a lot of our own internal resources, but when you're looking to go beyond that, you're looking for validation, you're looking for more genomes, like this is the place to be. Another thing that we're a part of is the AnVIL, and part of what makes it even more powerful over time is the NIH Cloud Platform Interoperability effort. And so the goal here is, like NHGRI has the AnVIL, other groups like NHLBI have Biodata Catalyst. Many of the same partners. There's programs like Kids First, and I know we have folks from those teams here as well, right? The Kids First Data Resource Center. That's the focus there is on pediatric cancer and birth defects. It's a very different type of data from a lot of what NHGRI is focused on. There are some overlaps. It's like, great, how do we make sure that those places where we're looking at rare disease, hey, that's a great place for overlap. How do we do that? When you want to talk to NCI and the Cancer Research Data Commons. How do we make sure that the cancer space really aligns? And so NCPI is looking to do that on the technical level. How do we make sure the APIs work, but also on the standards level from the data standpoint to say, hey, are we sharing in a consistent way using the same ontologies? And so it's an exciting effort to expand that impact. So it's not just coming to the AnVIL to get AnVIL data. It's coming to the AnVIL so you can use data from Kids First and from dbGaP and from Biodata Catalyst all together. So for a little bit of the who here, I think the three of us program directors are here up front. And so myself, Robert Carroll, we've got Jonathan Lawson here from the Broad Institute, and we've got Mike Schatz from Johns Hopkins University. And so we each are the prime awardees for the three parts of the AnVIL. And you can see a little bit what we're focusing on. And so we're very excited. We got in here. Do you guys want a brief intro? Yeah, okay. Yeah, so it's exciting. Each of us will get a chance to talk some more about the work that we're doing specifically. But we're very excited to be here. We're glad you're here as well. There's a lot of teams here. And so I do want to make sure that, hey, we highlight everyone. There's a lot of names up here. I can't read through everybody. But I think this shows the scope of what we're doing, right? And many of these people are more full-time engineers, right? Writing code, building tools to make things work. Many of them are just contributing a small portion of the effort as experts. Right, and that's part of why we're glad you're here, because we want to hear about your expertise and your needs as well. And we're really trying to bring together a group of people, again, very clinical. We've got a clinical lab director back there. We've got for clinical genomics, we've got people that are very computational. We have myself using EHR data for research. So we're interested in all those perspectives and want to make AnVIL a home for everyone. And I think we also wanted to highlight one team member in specific here. Jeff Leake, who was recently named one of the Times Top 100 in AI, and that's a team member working with Mike. And so it's just exciting to see these opportunities where the work that we're doing is really innovative, is driving the field forward in a way that's being recognized, not just in academic spheres, right, but externally of the power of what we're doing. And so I think this is really exciting to see. So I'll pivot a little bit talking about Vanderbilt's role in the AnVIL. And so we had joined on originally as one of the groups working with the Broads. There were 2 awards originally. Well, recently, we've been excited to be the AnVIL clinical resource in leading that. And so the goal here is supporting clinical genomics research and really helping move from a genome and basic science focus, really making sure that we can support the clinical and translational research side of what AnVIL in the cloud can be. And so we've got a number of pillars here that we're working towards. One of those is community and resource development, where this year we're honored to host the AnVIL Community Conference, but also doing things like developing workflows to support polygenic risk scores and making that available, right? And so the goal here is to say, hey, it's nice to have these computational tools, but do I actually, or the workbench, right? But do I have the right tools to use at that workbench and really supplying those? And so it's excellent there to develop more analysis pipelines. Additionally, we're working towards data portal integration. So we have representatives from I2B2 here as well, and say, how can we help make the data we have more usable to people? We have connections for REDCap. How do we help researchers bring their data more easily into Terra and the AnVIL for their own analysis or for sharing? In the clinical genomics research space, we've been doing a lot of work. Heidi and team have been doing a lot of work on clinician and lab engagement. So how do we get clinical genomes into a space where they can do research analysis? And development of the Seeker platform as well. And then finally, my team has been doing a lot of work on data standards. How do we make sure the data are harmonized and usable for discovery across settings? And so there's a big body of data here. How do we make sure that's very consistent and accessible and usable? And so again, a lot of different areas, and we're really excited to be doing this work. As mentioned, this is the second AnVIL Community Conference. We had one last year that was right next to the biological data sciences at the beautiful Cold Spring Harbor. I've never been before. It was awesome. Was really glad to be there. And we had some great keynotes from folks like Dashel Turner, and and a number of co-fests, which we're doing in this year. Opportunities for people to get hands on keyboard and kind of talk and work together. So we're replicating a lot of that here in a very different environment. Not as much water. But I guess it was very rainy, but it's a whole different setting. It was. I hope you like it still. Nashville's a great place to be. So we're just really excited to bring the community together again, and again, learn from you, share with you what we have at the AnVIL, and hopefully we can spark some new collaboration. AnVIL itself, by the numbers, there's other 24 consortia, over 277,000 participants with the data, 6 petabytes. I think we even have some news. I spoiled that I took it out, that we'll hear about in some of our top 10 in just a minute, about exciting updates on some of these numbers. Tons of tools and workflows through Whittle and Galaxy, with Terra, a bunch of public workspaces, hundreds of public workspaces and featured workspaces available to help show you work that's been done and published, or just to demonstrate what data is available and how you might be interested in using it. Galaxy, over 10,000 tools available. So we're really excited to have you here to continue to engage. And I think the thing of these numbers, I think NHGRI seems like they're still going to go up. We'll get more data, more genomes, more phenotypic data as well to analyze. And so we're excited to be a big part of that. So we're going to pivot a little bit into looking at sort of our AnVIL top 10 recent accomplishments. I don't know if it's actually 10 this time. I think we might have stretched it. That's why, hence the quotes. But some of the things we're most proud of here in the last year. And I'll start off, I think maybe taking credit a little bit for some of Jeff and his team's work. He's here as well. And this is about using I2B2 to support the eMERGE network in AnVIL. And so they've got a lot of data there that we've made available to them. I've done some work exploiting large language models to help support some of that transformation and mapping and understanding of the data. Working to deploy that secure data. I mean, security is hugely important to us, but to make deployment that's going to work for the eMERGE network. Working towards making that FSMA compliant for I2B2 to make it more available to more researchers through the AnVIL. And then working across data set querying. And I think this is one of the big things we really want to support, getting down into the details and making data more discoverable across queries. So that's what I2B2's been up the I2B2 team's been up this year. With Heidi and team on Seeker, they've done a huge migration to make it more performant. It's really neat to see what the tech can do when it scales up. They're looking to go live this month with this new backend. So you should see, if you're using Seeker, you should see improvements. If you're not, it's a great time to get in there and start using those tools. Improved their variant level matching with Seeker, looking at over 65,000 genomes. And that's a tool to help find similar or variants or individuals or samples with similar variants to what you're looking at, which can be huge when you're trying to understand rare disease, or what does this variant do? Are there other variants out there? Has this been seen before? So it's a great opportunity there. And they've continued to work with other labs to help bring data in. Again, I think this is one of those places where we want to help not just give the data that we've collected or that researchers contributed that are a part of the AnVIL, but to help you get your own data in a place where you can analyze it. And so that work with those clinical labs has been really important. Additionally, the development of polygenic risk scores. And so that's something that Matt Lebo and team have been working on, I think, and they have this is the work you're doing later that you're showing people. Yeah. So you can have a chance to do hands-on with this later at the ACC, but work building out the polygenic risk scores, tooling to support that and bring people and data in and generate the polygenic risk scores. So again, one-to-many conditions, it's a really powerful tool they've developed. And with my team, we've been doing a lot of work on harmonizing data and bringing up a lot of data that's been shared in the past, maybe with one field or just a couple of free text things. How do we bring that into a format that helps support discovery? So we've been doing a lot of robust data engineering practices using the security boundary of the AnVIL to be able to deploy this. And so, we've been building out things using DBT, for example, and some automation to actually transform data and make it more available. So we're working on, hopefully soon, you'll be able to see some of this data available in the AnVIL in this standardized format across studies. I think I now hand this over to Jonathan for some of the cool work they're doing. Thanks, Jonathan.
Thanks, Robert. So I'll give a quick update. Jonathan Lawson from the Broad Institute. Good to see a lot of you. Quick update on this. We'll hear a little bit more from my colleague Beth, who's here in depth. But an exciting announcement is we recently released the imputation service that combines the All of Us data set along with many data sets from the AnVIL. That came out in August. That reference panel includes over 500,000 participants. We're very excited about that. It's a significant increase on what has been out there in the world to date. So again, you'll hear more on that, but that's an exciting accomplishment. And also just want to highlight and say thank you to the All of Us team, many of whom are located here for their partnership in that. A lot of times NIH grants are not always funded for exactly what the future will be. So just a credit to them and to NIH folks on the All of Us and the NHGRI side for helping us make that happen. Another thing that we're excited about is self-service data ingestion. So a key part of the AnVIL that you've heard, we'll hear about, is the data. And we'll say more about that as the day goes on. But another thing we're doing to help those of you who are researchers who want to come in, use the data, use new data, find out about new data, is making submission of data to the AnVIL easier. So if you're trying to get data or you want to bring data to the AnVIL, that process is just getting easier and easier for you, meaning more data in people's hands faster. On that, to take up what Robert was mentioning that he had redacted, but is excited to share, from that presentation of the 6 petabytes that Robert mentioned just in the last 5 minutes, we're glad to announce that AnVIL actually hosts 8.4 petabytes of data from the slightly larger circles that you see here compared to the last slide. And an important note for you, again, if you're seeking to get data, is we are moving to a quarterly data release cycle. This has some operational benefits for our team, but it also means that you can reliably expect to receive a manifest of new data available from the AnVIL in the AnVIL every quarter going forward. So for those of you who are looking for new data for your next projects or to augment your existing projects, that quarterly release, you can see a sample of the manifest there, is going to come out each quarter. You can go to the anvilproject.org to follow that and make sure you get the latest updates on the new data in the AnVIL. And I think now I'm handing it over to Mike.
Thank you. Hey everyone, my name is Michael Schatz. I'm a professor at Johns Hopkins. I'm one of the co-leads for AnVIL. We focus mostly on scientific services, APIs, and outreach, critical and important parts of the team here. So of course, I'm excited about data. Most of my work over the years has been sort of a reanalysis of existing data sets, in addition to sort of leading the generation of new data. So I'm with you. Data's cool. But I think we wanted AnVIL, our ambitions for AnVIL were greater than just a static data repository. We wanted to have a lot of capability on board where you could do sort of primary analysis, new analysis of data, for any sorts of needs. I'd say our bread and butter are large-scale workflows. If you have, a cohort of 10,000 or 100,000 genomes, no problem. We can support that. We can help you through that analysis. We'll take some time and energy to do so, but we know how to do things at massive scales. And that's great when you already know what you need to do, and there's an existing workflow, you want to run with it. We'll show you. In fact, some of the trainings later this afternoon will tell you exactly how to do that sort of analysis. But in addition to kind of common things like calling variants or looking for GWAS or polygenic risk scores, a lot of analysis is like very new and dynamic. I think that's an exciting part about genomics, is that it's a very new field. Every day there's new questions emerge, there's new data types, there's new ideas, and we want to be able to support that as well. And then inevitably that means doing kind of really specialized custom interactive analysis. So in addition to our great support for workflows, we also have a variety of systems that are super accessible to be able to do interactive analysis. 2 of our pillars for that are Galaxy, which is a great system that provides tool access to more than 10,000 scientific tools. It provides sort of easy access to running the tools, building workflows, integrating with existing reference genomes, annotations for a whole ensemble of sort of custom analysis. One great feature of it is along the way, you can kind of, oh, I need to do this, and then I need to do that, and then I need to do this other thing. It'll keep track of all the provenance of everything. And then if you have 10,000 more samples you need to run it on, no problem. Just in one more click, you can go from 1 sample to 10,000. So we've been constantly integrating the latest version of Galaxy, making sure that's really up to date and hardened and available inside of AnVIL to be able to support your custom analysis. Another strong pillar inside of AnVIL is we have great support for R and especially bioconductor. This has become a very, very popular, important framework for genomics. If you want to do any sort of custom statistics or visualization, machine learning or processing, bioconductor is my go-to, right? There's just this enormous set of capabilities to be able to tap in to do that sort of very specialized custom analysis to support the goals of your own research. So again, we constantly partner with the team to make sure the latest and greatest versions of R are available, latest and greatest versions of bioconductor are available. There's also been a variety of specialized packages that have developed so that you can interface through the bioconductor world to tap into workflows or tap into some of the data tables so you can sort of do your analysis very, very easily and efficiently. So it's if you can load a file in bioconductor, you can load it out of AnVIL, out of some of the data stores that are there. So we want to really support that, to support all of you. And then finally, what I wanted to really highlight, I'm not sure, I guess this drives, yeah. Finally, ultimately, the success of AnVIL depends not on us, but depends on you, the community. And what are the amazing analysis that you've been able to support? And we're part of, I think, this team, us as sort of internal AnVIL community members. But ultimately, it's, we're here to support all of you. And we're just super excited to watch over the field, some of the scientific accomplishments that have been reached, using AnVIL as our primary platform. We pay close attention to like Google Scholar and other sort of formal citations. A couple of years ago, we had a paper in Cell Genomics. We're very proud that it was, kind of the cover story for that issue. And that's sort of one way we keep track, where we look at, official kind of, citations to that initial sort of marker publication. But we also look beyond that. We look, other sort of examples where people are referencing AnVIL data, or maybe some of the AnVIL tools, or using it as a place to house some of the analysis or build some of the communities there. And we're just really excited to see that really strong growth curve. In the early days, we had the paper and we had the first citation and we celebrated, and the second, now we have hundreds and hundreds of these, and it's just really exciting. And I think ultimately that's going to be one of the key metrics. I do acknowledge it's a lagging metric, right? Science, from the birth of an idea into all the analysis and peer review and everything, it takes, I don't know, months or years. So it is a lagging indicator, but we're really excited to see the growth curve for all of that. Okay, so those have been some of our highlights. And I just wanted to highlight, AnVIL is not a static resource. We're constantly trying to add new features and new capability, also try to bring in new data. So now we're going to do is spend a few minutes kind of reviewing give you kind of a sneak peek at some of the upcoming features ahead of us. There you go.
Thanks, Mike. Yeah, we're just, I don't know, again, excited. We want to show you what we're up to. I've also realized, sorry, folks online, now you can see us presenting. Well, for better or worse. Okay, so on to the roadmap. I get the privilege of going first here. I mentioned some of the tools that we developed. Actually, you know what? I've got one thing. Sorry, it hit me while I was earlier. One thing I want to say, if you are a Vanderbilt and you're using some of the data in Terra to analyze BioView ST data, you're right there. The AnVIL data is right there. You just have to go request access. You can start to use it. It's the same set of tools, same space. That's where we are. So if you have questions about what that means, feel free to come ask me. Again, this isn't some far off thing that we don't have connections to. Again, at Vanderbilt, we do have the data is available in Terra. You can use it for your analysis today. So again, that's right next door. It's like one house, really, for the analysis. So yeah, there you go. Okay, on to the work we're doing, the new and innovative development here. So we're continuing to develop with I2B2 again, and on these emerge whittles. So we've got those resource development as well. So this emerge, something that emerge has been working on in the phase 4 of emerge is actual clinical deployment testing of polygenic risk scores. How can this work? How can this change clinical care? And those whittles we're making available for broader AnVIL use. That's a key part of what we're doing this year. And so those whittles are workflows that would allow you very easily to compute these polygenic risk scores, which is a great opportunity to start to study and understand and potentially even implement yourselves someday clinically. We're continuing to finalize and publish kinship analysis pipeline. Again, the goal here is to make these tools, these common processes easier. So why would I, try and figure out how to do this on my own stuff when I can go to the AnVIL, I have a workflow I can deploy, I click, I point it to my BAMs and it goes, or my VCFs and it goes. And of course, we're here with the ACC this year, and so we're excited to have you for that. With I2B2, we're continuing to develop what data models are supported with I2B2 and supporting the emerge consortium as well directly through one of our collaborations, but also to work towards making I2B2 available more broadly to the audience with controlled access data. And so that it gives us a really powerful tool, especially for that clinical data research, similar to the types of tools, again, we enjoy at Vanderbilt with the synthetic derivative, to have those tools available more broadly. So when you bring your data in, you're able to use the AnVIL data and you're able to leverage I2B2. So we're excited about that continued development. With the Seeker, there's continued work on those clinical lab collaborations. Again, this is about getting more clinical labs to say, here's the right APIs, here's the right approvals that you need in order to bring your clinical genomics, again, patients consented for research, but to bring that clinical genomics data, those sequences, into the AnVIL for analysis. So that's continues to be exciting. We'll continue to get more partners engaged there. With data sharing for Seeker and AnVIL, again, this is about supporting clinicians doing clinical genomics research in the AnVIL, getting them able to load those clinical exomes or genomes directly into Seeker and AnVIL. Also working with patients to share their own genomic data through Genome Connect. With the automated interpretation pipeline, again, this is how do we make it easier for you to analyze your data. So you bring those genomes in, now you can analyze it, supporting the ability to redo those analyses periodically, right? As we continue to learn more about variants, we find more variants, we find more associations with disease. You want to be able to go back and say, hey, this genome we never fully understood was happening. Is there new evidence 6 months, a year, 2 years later, to give that opportunity to make that more practical? And so that's another tool that Seeker is working on. Also working with new data types and annotations. There's a lot of annotation tools out there. There's a lot of ways to kind of study and understand what might be new structural variants, what might be, again, that knowledge about variants and how those might affect processes that lead to disease. Can we include more tools to make that available? And so those are all some of the areas that Seeker is developing. With the data standards work, I mentioned this before, we've been doing a lot of work in building the foundation within Terra to be able to do harmonization in a secure way, robustly with good data engineering practices. So we're going to continue to do that, and we're up to speed, running through data sets, getting annotations of data that wasn't done with standards, annotated with the right terms, the right ontologies, and incorporating all that data together to make it available. And this year we expect to make available to you, the research community, some of this harmonized data. We're working also to support OMOP data as another way. We really want to, I think it's very hard to say, oh, I'm just going to share this OMOP data set. I've got this consented cohort of clinical patients. No one can do anything with that, right? Oh, here's 100,000 rows of data with a bunch of codes. Like, it's very hard to deal with, right? But if you had a place like the AnVIL where, hey, we can validate that for you. We can pick up this data. We can make tools that make it useful for you and for other users. All of a sudden, there's a value add there. That's not just, it's very hard to get this data and nobody's going to use it, and I've got to do a lot of work to dump it in a place no one will see it. It's, hey, it's very hard to get this data, but I have a place I can put it that knows how to receive it and knows how to validate it, knows how to provide it to research users. So, we're really excited to continue to support that. We've also been working hard on data dictionaries and provenance, and some of this we're starting to collaborate with the Phoenix Toolkit team, also with REDCap, but other ways to say, let's help people describe their data. Let's make sure that when you bring that data in, we're not having to go back 10 years later with our clinical ontologists and say, what terms do we think they meant when they said type 2 diabetes? Or even worse, T2D. It's probably type 2 diabetes. What do we think? So we say, hey, great. When I bring this, when I build my REDCap, when I collect data there or my spreadsheet, I've got ontology terms listed. And so we want to make sure that that's, again, facile for people, that it's not a huge challenge. We're not trying to put enormous burdens on submitters. We really want to say, hey, how do we as a community better collect data, better annotate it, make sure that it's clear what we're doing. So it's not just useful to me and not just clear to me, but that anyone could come pick that up and understand my study, understand my data, and replicate it or use that to power new discoveries. So that's where we're working. We're going to continue to do the annotation and harmonization again. So we're working this from both ends, getting the historical data up to speed and saying, hey, how do we help make this not be a problem we have to solve again in the future? So we're very excited about that. I think I'll hand it back off to Jonathan.
Thanks, Robert. So a few other updates to share here that we're excited about. Earlier, I mentioned new data releases that you can sort of subscribe to and follow as those come out. Also, in an interest of keeping us honest in front of the research community, we're also very clear about the Terra infrastructure and what features we expect to have come out. So you are able to go to the Terra roadmap to see what we propose, and this is not just a static site, but you're able to actually click in, see details about it. You can be invited to test, kind of beta test features as they come online and they're in preview. You also can comment questions, thoughts, concerns, ask why certain features aren't there on those individual issues you can see in those swim lanes. And our product managers, once we're actually overseeing that engineering work, will interact with you on that. So just, we are here to be a resource for the research community, both at this conference. You're welcome to talk to us, and we can just keep that going through sites like this one and through anvilproject.org. Just to call out some things that have been kind of exciting developments recently so that you're aware of them. We've done a lot in terms of visualizing cost management and giving you cost management tools so that you don't have any surprise overruns, so you know exactly what you're spending. Again, these are, I'm showing these here. You can click into those all on that that Terra roadmap to read a little bit more about them, to go see them in the Terra infrastructure. We're excited also to share more tools through the Terra workflow repository. And the IGV has had a lot of visualization improvements over the last 6 to 9 months or so. And I think even more coming. I was talking with the engineering team earlier this week. So we're excited to keep expanding on that and do some enhancements. There's also a few others that are interesting that we're working on. One is the RAS integration. So if you're not familiar, just to kind of hand wave that, that is making sure that you will be up to speed with the latest security requirements and accessing NIH data through AnVIL. And also there are some really exciting kind of workflow retries and notebook use, just to make it easier for you to not have to restart each time. So look out for that, follow that on the Terra website. And back to Mike.
All right, thanks for that. So I think one of the pillars of AnVIL is being very forward-looking. I mean, the NHGRI, not to be cheesy, but the NHGRI motto is, at the forefront of genomics. And I think we really strive to embrace that spirit and push forward on kind of all the latest and greatest advances. So one of the capabilities that I'm particularly proud of, having grown up in the world of genome reference assembly, is that we really support all these new initiatives to be a more complete and more comprehensive resource for genomics. So of course we have enormous support for GRCH38, our favorite reference genome that is out there. But if you've heard of something called the T2T reference genome, the telomere-to-telomere reference genome, AnVIL was the home to a lot of that work, where we were able to demonstrate putting the assembly together, but then more so demonstrating how that new complete reference genome improves our analysis of human genetic variation for both basic sort of population science, but also for clinical research. Beyond sort of the single reference genome, we're also the home for a lot of these new initiatives to develop so-called pangenome references, where instead of looking at just one sample, we're going to have really high quality references from many, 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 many diverse individuals from around the world as a way to sort of capture common variation in the human population. So about 2 years ago was sort of the first release of the HPRC, the Human Pangenome Reference Consortium. That's a set of really nice publications where they announced this first draft pangenome. And that really, I think, really conclusively demonstrated the value of having this diverse collection as opposed to a single linear reference. Well, here it is 2 years later, and I'm very excited to say, well, that effort continues. And the second build of the pangenome is happening like right now. The assemblies are complete, and they're building a graph that sort of represents all this variation. There's a lot of studies that are happening right now on the AnVIL, sort of tapping in and measuring how that new pangenome sort of further improves our analysis. I think, if you sort of fast forward, I don't know if it'll be 5 years, 10 years, or beyond, but eventually I like to think that all sort of analysis will be sort of anchored in a pangenome as a way to sort of capture common variation. And then more so, the technologies that are being developed to support T2T genomes and support pangenomes, I like to also see that sort of translate into the clinic, where if you're having patients with complicated, rare variants and rare diseases, and we're just not making a lot of progress using sort of standard short read sequencing, these projects also spearhead this technology that can let us access the complete genome. And then if we have the complete genome and maybe also the complete epigenome, there'll just be no place left for these genetic variants left to hide. So I'm just very, very excited about how these technologies will improve basic science and also clinical science moving forward. In addition to kind of those individual sequences, we like to also stay at the forefront of kind of the modern, most sort of advanced technologies for finding variation and analyzing variation. For a long time now, we've had great support for a great resource called Deep Variant. It actually had an origin story starting at Broad and then was transitioned to Google with some of the personnel there. And that, through many, many, many benchmarks, both internally and then other groups like the FDA has done benchmarking, that has really sort of cemented that Deep Variant is one of the world's most accurate variant callers. It uses something called a convolutional neural network to kind of analyze the raw alignments and then give you very accurate genotypes associated with that. It's great. We love them. We work really closely with the Deep Variant team. There's been many projects that have successfully used that inside the AnVIL. But again, it's not a static resource. We're always looking to the frontier. Very recently, as in like a couple weeks ago, the company NVIDIA announced that they have a new suite of tools called Parabricks, which is where they've taken many popular alignment and variant calling and RNA-seq tools like BWA and Deep Variant and a bunch of others, and have turbocharged them to use GPUs to accelerate the analysis. So instead of having to wait many hours or days to do your alignments and variant colors, you can get it done in a matter of like a few minutes. It does require GPUs, but again, because we're working in GCP, we have access to all these GPUs. So not only will it go faster, because it's so much faster, it's often cheaper to be using the accelerators to get this analysis done. So that's a major win. We have efforts right now happening to kind of integrate these into AnVIL, so that will be available in, I don't know, the coming weeks. So stay tuned for that. Another system that we're really excited about, this comes from Illumina, the company themselves. Again, they have a turbocharged analysis suite where they can go from kind of raw BCL files and raw FASTQ files, do the alignment super fast, do the variant calling super fast, get you sort of harmonized resources super fast. That can use GPUs. There's also a further accelerator called an FPGA that further turbocharges this analysis. I think this is kind of going to be a really important pillar about the future, where in addition to kind of regular CPUs in your laptop and your data center, sort of tapping into advanced hardware like GPUs and FPGAs is going to be really exciting and important in the future. That will also empower things like deep learning and AI. Right? These are very, very compute-hungry technologies. And because we're able to do this in the cloud, we're able to tap into those, make that accessible to everyone. Another kind of pillar, like I mentioned, is we're all very excited about AI. I'm a professor. I'm teaching a class right now, and 100% of the students are using ChatGPT on every assignment. I think that is just the reality of the world that we're moving into. At OpenAI, Andrew Karpathy sort of made this bold statement that English, natural language, is the new programming language. And I also have been personally having a lot of fun vibe coding in programming languages I don't really even know. But it just makes programming super accessible, where I don't need to know the syntax of this library or that language. I can just express in English what I want to happen, and then the large language models are really good at translating that into compute code. So we're really excited about tapping into that capability in AnVIL more broadly. Right? I think some people are really proficient at the command line and really proficient programming, but no one is born with that. That's a skill that we all have to develop. And the reality is we all have so many things going on. Do you really want to commit your 10,000 hours to learn this one programming language for this one analysis? Or could you take advantage of these new technologies that tap into it? So yes, sometimes it's not that programming is going away completely, but I think it makes it a lot more accessible, a lot more democratic, and just a lot more engaging and powerful for end users. So as a demonstration for this, one of the key pillars, as I mentioned, one of the key pillars of AnVIL is our technology Galaxy, which has a really rich API where there's any sort of analysis you want to do, look for data, look for tools, run workflows. There's a great API there. It uses something called that Fast API, which is like a very sort of popular standard for this. On top of that, we've written what's called an MCP server, a model context protocol server, where now in a natural language like English, you can just sort of talk to it, and then you can do all this very sophisticated analysis. So if you're a wet lab researcher, you might this is now a scientific question. I have RNA-seq data from mouse liver samples. Help me analyze differential expression. Like at that level of high level, it will then translate it into finding these specific data sets, identify the specific tools, specify the exact workflows, kick it off for you. There's still going to be a human in the loop, right? Of course, these large language models are exciting and can do amazing things, but they're also very uneven in that they will sometimes hallucinate and do weird things. So it's really important that we're doing this in a system where it's totally transparent what the analysis was. It's really important that it's all the provenance is collected. It's completely reproducible. You can go inspect it. You can go unwind when it does weird things, go back to where it's solid, and then pick it up again. So we really are excited about embracing these technologies moving forward. We have kind of like an early demonstration of this. Realistically, this will be, I don't know, several months or a year out before this is more generally available, but we're really excited about embracing these new technologies. And furthermore, with our partnership with Bioconductor, again, to support that sort of interactive analysis where you need to do the custom statistics or machine learning or whatnot, we have a lot of great resources that are available now and then moving forward. Some of the more sort of user-facing ones, Bioconductor has like more than 2,000 packages available, and it's just like really hard to stay on top of what's there. So we're building out all these sort of curated resources that sort of guide people to, if you need to do this type of analysis, here's a great protocol. If you want to do this type of analysis, here's a great protocol that can really sort of guide you and walk you through all the analysis there. So already there's several of these that are available, and then we're just building out these community resources to kind of grow and help other people. To support that, behind the scenes, there's a lot of new technology to help you make these monographs. I think one of the strengths of Bioconductor is that you often get these like vignettes that say, oh, here's this new package, and here's a little short example of how to use that. And we're trying to expand on that technology to make these sort of monographs even more efficient, easier to write. Again, it's all about making all this exciting tooling more available, more generally powerful and accessible to everyone. So look forward to that. And then again, similar to what we just heard, because we're running in the commercial cloud, we can tap into all these great accelerators like GPUs, like FPGAs, like TPUs, and other hardware tech accelerators to really tap into the latest and greatest models and make it really powerful. And then another sort of thing I want to highlight is just we're constantly trying to enhance the user experience. In addition to the great enhancements that Jonathan was talking about in Terra itself, we're constantly trying to optimize these experiences here, try to make it faster to boot up into them, try to make it less expensive, right? Because we are running in the commercial cloud. We're always sort of conscious of this. Trying to make it sort of simpler to get data in and out, and just really trying to streamline the user experience. So a couple of things that we're highlighting here is we're sort of kind of doing some engineering work behind the scenes in Galaxy to really sort of make it a lot more dynamic, and this will reduce costs and just sort of improve the user experience. For our bioconductor, again, we're trying to do this hard software engineering to shrink down the footprint. So again, it's faster to boot up, be cheaper to run, and just give you a much more optimized experience. And we're also trying to simplify some of the environment choices that are there. In addition to kind of those components, we also work really closely with the GA4GH and some of the standards that they've been working to develop. There's a great standard called TEST that is emerging for executing tools. And we're working really closely with that community there to kind of harden this. And I like to think of AnVIL as being like a premier example of the power of GA4GH standards, where now we can have great interoperability across many different consortiums, many different tools in this ecosystem. So we're really excited about that. So there's always constantly new features being announced and new technologies and new data sets. We just heard, we're moving into this like quarter release. So every 3 months, expect massive announcements about the data that are coming. So if you want to like really stay informed about this, please subscribe to our like LinkedIn feed. That's probably our sort of preferred venue for that. We also have accounts on Blue Sky and a few others, but LinkedIn realistically is a great place to do there. I just see that emerging as like a really nice sort of even level-headed environment to have professional discussions. So that's what we're really striving for. So please stay tuned for that. And then throughout the year, there's a number of workshops that we offer. A number of demos that we offer. We offer monthly office hours. So you have questions, you can kind of hop on a Zoom call, come talk to some of the developers. So we're trying to have this like multimodal strategy where if you have questions, you have problems, you have new resources you want to highlight, we're trying to empower you, the community, to sort of tap into us and discuss all of this. And then finally, last thing I wanted to highlight was, as we heard, we're in a great place now where we have really optimized the process to sort of ingest new data sets and make them broadly accessible. So if you are working on a project and you have a data set, be it, 1 sample or 100,000 samples, and you need a home for that, please come talk to us. If your work is sponsored by NHGRI, we can really help you support that and really help support some of the costs associated with that. If your work is not supported by NHGRI, come talk to us anyway. We can help you navigate some of the systems. There might be some strategies that we have there to help support this work. Okay, so that's it for a couple of highlights from the upcoming roadmap. Okay, well, great. I think we're like exactly on time, which is always really fun to see.